and all the infectious diseases and zoonotic diseases. So this is the main reason, this contributory effect. Also, if you look at the pets, yes, as I told you previously, pets has not been included for the biomass statistics previous slide, but the pets, it's also very common, and it's our social counterpart, because most of the people, they would like to have pets, even kids, and there's no different adults or maybe mature people, but still they would like to have pets. The dogs, yeah, this is actually statistics from US. The dog is the most popular pet, of course. If you get the world statistics, there are 471 million dogs are kept as pet globally. So we cannot forget about the role of dog in our social context. That is why I'm going to highlight in this slide. And this friendship is not the very recent one. We have been, if you look at the prehistoric cave painting, of course, it was, we could find evidence that our strong friendship, that long years friendship. That means we cannot forget about dogs and humans. So that is why the One Health come to the picture, because we have to, if you consider the healthy people, of course, we have to consider the healthy dog population as well. So I'm going to talk about Staphylococcus pseudintermedius. I think being my audience is mostly from veterinary background, so they know the organism well. But if I go into brief a little bit, which is gram-positive cocci, and of course the COVS-positive bacteria. The problem sometimes we face in human medicine, this organism is really misinterpreted as Staph aureus, because they know really the Staph aureus, but a little bit information about Staph pseudintermedius. If you get the colony morphology, which is gray colored and uneven medium-sized colonies, you could see in the next slides. And the other important thing is having the double zone of hemolysis, because it's a typical hemolysis is usually we can see on blood agar. But different biochemical tests, like you could use whatever the technique to isolate or diagnose the condition, like conventional methods, start with gram stain and followed by series of biochemical tests, and end up with sugar fermentation test also we can use. But molecular methods can be used, like PCR, qPCR based method, or otherwise we can have the monitor of MS to diagnose the Staph pseudintermedius. This is the picture of these grayish color colonies, and the other side actually you could see the hemolysis pattern, which is really a good characteristics to suspect as Staph pseudintermedius in your culture. And if you get the Staph aureus and Staphylococcus species, the actually people, they came to know about Staph aureus in late 1890, and maybe 150, maybe 130 years back. But Staph pseudintermedius, which came to know around 20 years back, it might be the reason that some of the people in life science, they don't have much aware on that organism. But compared to Staph aureus, is the most popular organism. Staph pseudintermedius is, which is also coming up in epidemiology, we could discuss it. So in the under One Health concept, the people in the whole world now, they are talking about this concept, because if you look at if you really focus on healthy people, of course we need to have the healthy animal and healthy environment. So without having a healthy environment and healthy animals, we are not in a position to talk about healthy people. So dogs here, actually we are talking about the healthy animal. Is our animal is our dog here, but and also some environment factors as well. So the same things I'm going to summarize here. The dog is there, the Staph pseudintermedius, which is commonly found in dog, but not only dog, but there are some other hosts like horses and cats and some other pigeon and other organisms, other hosts as well. And it's a human and also the environment. So we are going to have this little bit discussion on this dog, human, and environment during my presentation. And the other important things that, because this is actually, this slides was actually, this table was taken from one study published in 2012, quite a little bit old, but It's not only Staphylococcus, but with that there are some other Staph aureus, Staph epidemis, and what I'm going to say here. There are some other bacterial species as well, almost more or less similar to Staphylococcus. That is why we need different tests and a series of tests to differentiate Staphylococcus from Staph aureus or from maybe from other bacterial species commonly found in dog. And the if you little bit discuss about the sequence divergence of Staphylococcus with the other related organism, of course the differences are there. Here they are a little bit closely associated in the phylogenetic analysis, but of course you could find any differences, because that's why it is considered as one species, but they are closely associated with the sequence divergence in Staphylococcus. And I was trying to say the diversity of Staphylococcus. If you look at the same species, if you go here, actually the diagram from pulse field gel electrophoresis, but that is my take home message here, the diversity, because even the same species, you could see very really differences in Staphylococcus, because this diversity is a key factor in Staphylococcus. So I'm going to talk about the Staphylococcus a little bit in dog. So if you look at the epidemiology, Staphylococcus, it is commonly found in healthy dog. So it can be 42%, 90, maybe 40 to 90% percentage of, but this percentage is really different based on the technique. Maybe it's depend on a sampling type, whether we are going to get a sample from pharynx or anal swabs, and also number of samples per animal. Maybe sometimes we have to get multiple samples, more than one sample per animal, and also some isolation techniques. So the percentage is varied with all these factors. So we cannot say which one is right and the other one is wrong because of considering all these factors. But the organism is basically adapted to the family Canidae. That means a canine family. But the recent and also reported in other hosts like cats and horses, and also I have seen the publication in cows. And if you look at the transmission, of course, the horizontal transmissions is there within the same species, like within the canine host, transmissions can be reported, and also interspecies transmission. I mentioned from dog to cat, or maybe dog to human, and the zoonotic transmission, of course, the transmission from animal to the human. Other important thing we have to keep in mind, the Staphylococcus is part of the normal flora. That means it is there. We cannot eradicate it because it's a part of the microflora, normal flora. But it can be opportunistic pathogen, but we don't know most of the factors associated with colonization. Still, we need a lot of studies and things had to be investigated, but it's opportunistic pathogen. So, and the other importance as veterinarians, because the clinical importance is quite important for us, like it is the most common cause of dermatological infection in dog. For example, the canine pyoderma and some external ear infection like otitis externa. And the other thing is, it's the second most cause of urinary tract infection in dogs. So that's why the small animal medicine and companion animal medicine, the Staphylococcus is quite important organism. And this table in your left side, so I extracted from one of the article, that is the prevalence of bacterial isolated from pyoderma and otitis externa in dogs. So actually I was trying to say here, because Staphylococcus not only is there, yeah, Staphylococcus is there, but with that, there are a lot of other bacterial organisms can be found, like maybe Pseudomonas and Proteus. It's always the multiple bacterial involvement is there. So that is why the
I think we lost the. Let me try to see if I can.
So that's why we have to have the right sample at the right sample, the location. And also this is actually the graph from the MLST database, pseudintermedius. If you look at that, most of the publication and